So for this next section, now that we've talked about replication, we're going to talk about gene expression, or the flow of genetic information within the cells. If you remember the central dogma, we've talked about it several times before. DNA will be used to create RNA. RNA will be used to create proteins. Okay? This is known as transcription, while this is known as translation. Okay? If you need help remembering, they are in alphabetical order. So using DNA to create RNA is transcription. Using RNA to create proteins is translation. When you do both and actually move all the way through this process, it's known as gene expression. So a gene is expressed, okay, when it is being turned into a protein. Right? So let's talk about transcription first, the first part of gene expression. Okay? Now, in transcription, the lucky part of this is that we are utilizing a single enzyme. Yay, it's about time. I know you guys are used to us dealing with lots and lots of enzymes. Okay? So RNA polymerase is the enzyme responsible for transcription. Okay? So RNA polymerase, its entire job is to convert DNA into RNA. Now, be aware when we use that term conversion or convert, we're not saying that the DNA becomes RNA. We're saying the DNA is used as a template to create RNA. Okay? So, the template strand is the strand of DNA that we will use as a template to synthesize a strand of RNA. Okay? And therefore, it will also be used to direct the production of a protein. Transcription has 3 steps. Okay? Initiation, basically starting. Elongation, adding individual nucleotides together to make a long strand of RNA. And then eventually termination. So in simple terms, we are going to start, make it long, and then stop. Okay? So, how does this work? Well, the DNA sequence called the promoter is actually where the RNA polymerase binds. So the polymerase binds as a promoter, at a promoter, and then once the polymerase is there, it actually wedges apart the 2 strands of DNA to open them up. Okay? The DNA polymerase then begins to unwind the strands of DNA and starts adding on complementary RNA nucleotides. Okay? This elongates the strand. So as those nucleotides get added, the RNA strand gets longer and longer. You can see it here. Okay? Now, eventually a terminator sequence will be reached, and this signals the RNA polymerase to actually fall away from the DNA. So it detaches the RNA polymerase. The fancy term we use for this is that it dissociates. Okay, that the RNA polymerase dissociates from the DNA, and the RNA itself dissociates from the DNA. The DNA coils right back up, okay, as if nothing has happened, and the RNA is then free to be used to create a protein. Okay. Now, this is going to look a little bit more, okay, like, we'll go with purple. Okay. This is probably going to look a little bit more like this. Okay. When it comes to the DNA, you gotta understand there will be 3 parts to the gene that will be used. That's why this is gene expression. This is the gene, the piece of DNA that will be used to create a piece of RNA. Okay? It will include a promoter sequence. This is where your RNA polymerase binds. Okay? The gene itself, okay, which will be used as a template for RNA production. Okay. And then the terminator sequence, which is your great big signal to stop. Okay. So we see if I can animate this as best as humanly possible. Okay. Obviously, there's some limitations to what I can do. Okay. So the RNase comes in, binds at this promoter. It begins to travel down the gene where it creates a long strand of RNA until it's the terminator sequence, okay, and dissociates away from the gene. Now, in this instance, where the promoter binds, or I'm sorry, where the RNA polymerase binds at the promoter, forcing those two strands of DNA apart will actually create a bubble. Okay. This time, we refer to this as the transcription bubble. Okay. Because remember, a major part of being able to use the DNA is going to be forcing those two strands apart. Single-stranded DNA is usable. Double-stranded DNA is protected. Okay. So double-stranded DNA is great for when it comes to protecting the DNA and keeping it from being broken apart. Okay. But in order to actually be able to use it, you've got to get it single-stranded. So these temporary little bubbles get created that eventually come right back together after the RNA is made. So your book does a decently good job of this, explaining it in three terms. If you want to follow along in your book, it's Table 8.3, initiation, elongation, and termination. Okay. Now, when it comes to the RNA that's being made, RNA is going to be similar to DNA in terms of its general properties. Structurally, though, it's going to be quite different. RNA will be single-stranded because its goal is to be used and read very quickly. RNA eventually degrades, so double-stranded RNA is not something we see in animals, okay? Or pretty much in any type of cell, mainly because then it's not going to be useful. We want the RNA to be usable. Okay. If it's double-stranded, it's protected RNA, it's not going to be used. Now, there are some applications to that, but that's an entirely different type of lecture. Okay. RNA, interestingly enough, can assume secondary and tertiary levels of complexity. In other words, RNA can fold. Okay? And RNA can be three-dimensional. And if we haven't learned anything else, it's that three-dimensional structures have functions. So we actually do see functional RNA. More specifically, tRNA and rRNA are folded RNAs that I like to call machinery RNAs. These are good working RNAs. They have jobs. They do things. Okay. RNA will contain uracil, okay, as a nitrogenous base instead of thymine. So there is no thymine in RNA, and ribose instead of deoxyribose. So what that means is that if your DNA strand, and I shouldn't even have to tell you that this is a DNA strand, just looking at it and seeing those thymines means that it's DNA. Okay. The complementary RNA strand that goes along with it should read. Try it at home. Hopefully you are saying something along the lines of U, A, G, C, A, U, U, G, C. Okay, so this would be your complementary piece of RNA. Okay, this is what you're going to be using to make a protein. Or it can be folded and turned into tRNA or rRNA, depending upon the needs of the cell. Okay. So, three main types of RNA that we're going to utilize within cells. Messenger RNA, this is your information derived from DNA. This is the gene. This is your instructions on how to build a protein. It basically tells us which amino acids to add and in what order. Okay. Transfer RNA. Transfer RNA is going to carry amino acids to ribosomes for the production of specific proteins. Okay. And the transfer RNA does this based upon the information found in the messenger RNA. Okay. Ribosomal RNA, whose job it is to help maintain the shape of the ribosome. So ribosomal RNA helps maintain that three-dimensional structure of the ribosome. If you remember, ribosomes are protein-producing factories. Okay? That's where we're actually going to make proteins. So all three make up a necessary part in the next step of translation. So, translation, that second part of gene expression. Okay? Now, a couple of notes before we dig a little bit deeper into this. The ribosomes of prokaryotes and eukaryotes are different sizes. Okay? This has been mentioned to you before. It will come into play in this chapter as well as in the next test. Remember this, commit it to memory. Okay? Prokaryotes, that 50S large ribosomal subunit and a 30S small subunit together making a 70S total ribosome. Okay? So remember, odd prokaryote, they get the smaller numbers and the odd numbers. The eukaryote, even eukaryote, a large 60S subunit, a small 40S subunit that together make up an 80S ribosome. Okay? Now the large and the small subunits actually have different jobs. Okay? The small subunit's job is to bind the 5' end of mRNA. Basically, the small subunit is responsible for making sure the mRNA moves through the ribosome and doesn't get kinked. Okay? Think about it like trying to thread beads through a hole. It's going to make sure that as these mRNAs move through the ribosome, that they don't get kinked and so that they're read correctly. Okay? The large subunit is responsible for supplying the enzymes and the sites necessary for bringing tRNAs into the ribosome. So if we're putting the appropriate tRNAs into the ribosome, that's what helps link the amino acids to one another. Okay? So the large subunit is responsible for the enzymatic reactions that help read within the ribosome. Okay? Now, if you look at images of a ribosome, one thing you'll notice is these A, P, and E sites. Okay, all three of these sites are very important to producing a protein. What you're going to see happen is the mRNA moves between the small and the large subunit, and these tRNAs are actually going to have matching complementary nucleotides that go along with the mRNAs. And the tRNAs and the mRNAs are going to match each other, complement one another, and that's what makes sure we have the exact right nucleotide, the exact right amino acid added to the growing strand of amino acids. So this makes sure we put the amino acids in the correct order. There are two spots for tRNAs to actually dock within the ribosome, the A site and the P site. Okay. Once the two tRNAs are in the A site and the P site, the amino acids can be attached to one another through a peptide bond. Okay. And then this tRNA in the P site moves out of the ribosome to the exit site. The tRNA that's in the A site moves over to the P site. Okay. And a new tRNA comes in and takes a spot on the A site. Okay. So I always tell people we ape our way through the ribosome. A site, P site, E site. That's how the tRNAs get it. They ape their way through the ribosome. Moving from the A site, from the P site to the E site. And the E site is an exit tunnel. Okay? The tRNAs don't sit in it. It's a way to get out of the ribosome to make room for new tRNAs coming in and carrying new amino acids. Okay? Now, how does this work? Well, it's highly dependent upon the genetic code. Okay? The fact that mRNA is read in groups of 3. Okay? So there are groups of 3 nucleotides that actually dictate which amino acid is added to the growing peptide chain. And each group of 3 nucleotides is called a codon. Okay? This is the codon chart out of your book. Okay? And what you'll see is that each group of 3 nucleotides codes for putting a specific amino acid in place. So the mRNA is read in groups of 3. Okay? Now, you need to be able to read a codon chart. The one that is included in your notes is the exact one that I use on the test. So a quick little refresher on how to read a codon chart. Okay. So first position. Okay. This is the first letter within the codon. So if our codon is, let's go with, we're going to make a simple one, A. Okay. This is first base position, the first letter within the codon. So if you look right here, A. What that means is that everything in this row, okay, starts with A. It's A something something. A blank blank. Okay. So we've taken care of that first A. Right? So let's look. Second base position. Okay, you see A, right? What we're saying is that everything in this particular column is blank A blank. Okay? Well, we want to know where A is. So A means that we're going to be somewhere in this particular box. So we've narrowed it down to the box. Okay? If you notice, these are missing on the codon chart that I use. They will not show up on the test. So be prepared to be able to read the chart that you have in the notes. Okay? Now, the last portion of this, okay, has to do with this third base position that you see over here. And if you look, third base A, it's the third one down. A. We go all the way over. Okay, look what we hit. Lysine. AAA codes for the amino acid lysine. There are several practices, okay? Several practice options for this online, and you can ask for more help or answers to those at any point in time. Okay? Now, another thing that you might have noticed is that there are 64 possible codons. Okay? 64 possible codons. There are only 20 naturally occurring amino acids, so the codon chart is what we call redundant. Okay? In other words, there are several instances where several different codons will code for the same amino acid. If you look right here, okay, all 6 of these groups of 3 all code for leucine. Okay? So there's a lot of redundancy. Leucine seems to be a relatively commonly used amino acid. Okay? This allows for the insertion of the correct amino acid even when sometimes mistakes occur in the DNA sequence, because then there's no real difference between CUU and CUC. Okay? This is something that we call the wobble position. If you notice, I'm going to go back one more time. The thing that doesn't seem to matter the most will be that third base. So CC anything is proline. AC anything is threonine. GC any of the 4 is alanine. Okay? So that third position is what we call the wobble position, where only those first 2 nucleotides are really required, okay, to get the correct amino acid. This lets some variation or mutation happen without actually changing the amino acids that are put in a specific order. And we know how important the order of those amino acids are, because that's what gives us tertiary and quaternary structure. It's what allows the proteins to be 3-dimensional and have their function. Form dictates function. If we can't get these to fold correctly because they don't have the right amino acids, okay, we can't get the proteins to function correctly. Okay? So if you want to go through this step by step in your book, honestly, I'm going to recommend another YouTube video that's going to break it down slightly more easily. This particular diagram is relatively wordy. I would pay more attention to the pictures, the images specifically, than I would the individual text. Okay? Now, one thing that is important in here is that you understand, and I'm going to flip back, okay, and get rid of some of these, is that when it comes to codons, there's one very specific start codon. Okay? AUG codes for the amino acid methionine. That's the same in every type of organism. I will, however, tell you that methionine in prokaryotes is slightly different than methionine in eukaryotes. It is still, however, methionine. Okay? This is the start codon. It is constantly where we will begin adding on amino acids. So you always look for the first AUG. That's codon number 1. Okay? In any sequence, the first AUG is codon 1. It doesn't matter if there are 5,000 C's and U's before it. Okay? That first AUG is codon 1. Okay? There are also 3 stop codons. Okay? UGA, UAA, and UAG. Okay? All of these have no tRNA pair. They have no tRNA complement to them. What that means is that they leave a space open on the ribosome after the tRNA moves into the P site. A protein slides its way into the A site very quickly and causes the ribosome to dissociate from the RNA. This signals the end of translation. Okay? No more making of the protein. So you can see it here. Okay? Us adding amino acids onto one another, filling up that A site and the P site. Okay? Continued moving. Okay? Here's our tRNA moving down. Okay? Moving into the A site. Okay? And then a new tRNA coming in. Another tRNA coming in. Eventually, once the stop codon is hit, okay, this space will be empty. A protein will jump in and cause the entire thing to dissociate. The entire ribosome and the RNA strand and the protein to dissociate from one another. After this, protein folding can begin. So I'm going to end this particular lecture with translation. We'll talk about differences between prokaryotes and eukaryotes and a little bit more sort of mastery of the genetics and some differences that we find with prokaryotic organisms in the next lecture.